Hello everyone and welcome to the granulation of powders experiment. This is the practical part. Always clean thoroughly all equipment, tools and working area before and after working. And always remember, many of these steps should be done parallel. First, pass enough large quantity of lactose through a 1.4 mm sieve to remove any lumps or aggregates. You may also use 1.7 mm sieve. Now, using a tin paper or aluminum foil, weigh 400 grams of the sieved lactose and transfer to the kneader. Again, carefully transfer the weighed quantity to the kneader. In this video, we are showing you the use of water as a binder. The same procedure should be repeated with other binders. Start paste preparation by adding the binder. We will start, as I said, with distilled water. Add it portion-wise. How? Using a 10 ml measuring cylinder, add several additions, each addition of 3 ml of distilled water every 3 seconds to 1 minute. Each time add it to different place. After each addition of the binder, cover the chamber and operate the kneader with medium velocity for 30 seconds to 1 minute. Always be ready, such that between additions, always prepare the 3 ml portion for the upcoming addition. After the third addition and massing for almost 1 minute, turn the kneader off using the emergency button and test the mixture for the end point. The end point we are aiming to achieve is prior to paste formation. Again, the required end point is the point prior to paste formation, which can be measured by taking a mass from the mixture using your hand and squeezing it between the palm and fingers, then hitting it with other hand index finger. If the squeezed mass broke down completely, continue the addition of the binder and check for the end point using the method we mentioned. After each addition, till the end point is reached. In the shown example, after the third addition, the mass broke down and we didn't achieve the end point, so we added a new portion. If it remained coherent and only broken from the peripheries, then the end point is achieved. So return the mass to the kneader and perform massing for extra 1 minute to ensure homogeneity and to get rid of any large agglomerates that might be formed. Here is, in the example, the formation of a coherent mass that is only broken from the peripheries. Now, we will return the mass to the kneader and perform massing for extra 1 minute to ensure the homogeneity and to get rid of any large agglomerate as we said. This has been done without the addition of any quantity of the binder. Remember, you can use a spatula to remove any sticky particles or paste to the kneader walls. And now, after the extra 1 minute massing, we will transfer the paste to an empty clean tray. Then, after achieving the end point, transfer the wet mass to an empty cleaned tray. Use a spatula to completely remove the mixture from the kneader chamber. Again, after each addition, after the third addition, we need to check for the end point until we achieve it. You may need some time to remove the paste, all paste from the kneader chamber, so try to make it quick because it might be the rate limiting step. Make sure that meanwhile you are removing the paste from the kneader chamber, that one of your colleagues is preparing a clean, empty, weighed tray. Record the weight of a tray during this time. Again, the prepared tray should be weighed and the weight should be recorded. Make sure to label the tray with the binder name. Allow the paste to pass through the sieve fitted to the granulator, oscillating granulator, and collect the wet granules from the granulator on a pre-weighed clean tray, the one you prepared, and spread to form a thin bed of granules to assure uniform drying, complete and uniform drying. Weigh the tray containing the wet granules and record its weight as wet weight. Now, place the wet granules in a preheated oven at 7 degrees Celsius for 45 minutes to allow for complete drying. Meanwhile, prepare the other two binders. Sucrose solutions 50g% weight per volume and starch mucilage 7.5g% weight per volume. These can be prepared again by one of your colleagues. Meanwhile, you are preparing the paste with the first binder. To prepare sucrose solution 50g% weight per volume, place 50g of coarse sucrose in an Erlenmeyer flask. Then add distilled water up to 75 ml or 80 ml mark. Continuous stirring will be needed. You may use magnetic stirrer hot plate to accelerate the process. You need to use the magnetic bar, the small white capsule you are watching. When sucrose completely dissolves, remove the magnetic bar and complete the volume with double distilled water or distilled water up to the 100 ml mark. Mix well and use in the same manner you did with distilled water when used as a binder. Again, repeat the same procedure with another 400 grams of sieved lactose, but this time using sucrose prepared as sucrose solution 50g% weight per volume as the binder. To prepare starch mucilage 7.5g% weight per volume aqueous paste. First, weigh 7.5 gram of starch. Disperse the 7.5 grams of starch in up to 100 ml of distilled water in an Erlenmeyer flask. Make sure to complete the volume immediately. Then put the prepared dispersion on a magnetic stirrer hot plate. Use a magnetic bar inside the dispersion. Continue warming the dispersion with continuous stirring until a translucent paste is formed. Remember that heating is essential in preparing the starch mucilage. Here is the translucent paste. Repeat the previous procedure with another 400 grams of sieved lactose, but this time using starch prepared as starch mucilage 7.5 gram % as a binder. After the time elapsed, after 45 minutes of drying in the oven at 7 degrees Celsius, remove the tray containing the dried granules from the oven and wait to allow it to cool down. Then record its weight as the dry weight. Now allow the dry granules to pass through the sieve fitted to the granulator. This is called the regranulation procedure step or dry granulation. After the regranulation step, put the granules in an appropriately labeled plastic bag. The label should contain the section, the group number, the binder name, and the granules name. For example, granules prepared using distilled water as a binder are named granules A. Reserve these granules in a dry, dark, closed place to be tested in cycle 2. Again, granules A, B, and C prepared in this experiment will be tested again in cycle 2, experiments 1 and 2. So you should reserve or store these granules in a dark, dry, closed place. Special thanks to Mr. Amid Al-Tahir and thank you all for watching.